Okay, so that leads us to the next topic, which is how the mechanical and the chemical forms of digestion interact in the small intestine. So, the mechanical digestion in the small intestine is promoted by a process which is called segmentation. So if peristalsis is the propulsion of the content of the GI tract in one direction, segmentation is pretty simple, is separation of the content into separate parts. And this is not mostly, it's completely caused due to the contraction of the circular smooth muscles of the small intestine. When the circular muscles contract, the lumen diameter decreases, leading to the separation of the content of the small intestine, known as chyle or chylo, or what we call the mix of chyme and digestive enzymes, the chylus, into separate parts. And the advantage of that is that the segmentation leads to an increase of the surface to volume ratio. So by increasing the available surface for the digestive enzymes to act, they act more efficiently, more effectively, and faster. So the strength of the contraction is regulated by the food content and by the food amount. The peristalsis, of course, is the same as the one that occurred in the esophagus and in the rest of the GI tract, is due to a combination of the contraction of circular and longitudinal muscle, and the coordinated contraction of the circular and the longitudinal muscle generates an area of high pressure behind the content and an area of low pressure in front of content. And so the peristalsis initiates with a segmentation movement, so with the contraction of the circular muscles behind the chyme, the bolus, or whatever it is, depending on the segment of the GI tract where we are, in coordination with the contraction of the longitudinal muscles. So when the longitudinal muscles contract after the circular muscles contracted behind the bolus or the chyme, that will generate the propulsion of the content into one direction. So this, of course, has the main role to push the content into one direction, but also during the peristalsis, the content of the small intestine gets mixed up. And it gets further mixed up with the digestive enzymes, increasing the efficiency of the chemical digestion. And the frequency of the peristaltic contraction varies in the different regions of the intestine along its length, actually. So with highest frequencies in the duodenum, a little bit less in the jejunum, and the lowest in the ileum. So on top of the digestive and absorptive functions of the small intestine also has an immune function. So the bacterial load in the small intestine is fairly low, as we've seen. This is due to the fact that the small intestine epithelium generates bactericidal peptides, such as the lysozyme. But even though the bacterial load is low, the chyme is actually close to sterility because of the stomach acid. The problem is that when the chyme enters the small intestine, the neutralization of the acidic pH due to the bicarbonate will lead along the length of the small intestine to a progressive increase in pH until close to normality, to neutrality. So that increases the risk for bacteria to grow. Okay, because you have to imagine an environment full of nutrients at 37 degrees and with a pH which gets close to neutrality when the chyme moves away from the stomach. So it is very important to actually keep the level of bacteria low in the small intestine. And there are several strategies, one of which are the antibacterial molecules I've already talked about. It's mostly generated by the pannate cells. But also the motility itself of the small intestine promotes the movement of the content of the small intestine. And, by moving things around, actually moving things, from the oral side to the rectal side of the small intestine, that decreases the probability for infections to arise and for the bacteria to grow. The mucus itself is not a good growing medium for bacteria, mostly because of the presence of antibacterial molecules. But also, if these mechanisms would not be enough, the small intestine contains lymphoid follicles and lymphocytes which secrete immunoglobulins. So an adaptive form of immunity which keeps the small intestine clear of pathogens. So on the function of the small intestines, of course, we need to talk about the two main accessory glands, the liver and the pancreas. Now, I'm not going to talk about the liver in detail because you have already talked about the liver in great detail in Dr. McNish's lectures. So I encourage you, to look at Alistair's lectures and to connect them to the content which has been delivered here. Today we'll talk about the liver mostly in relation to its digestive function. Amongst the other several functions of the liver, one of them is to produce bile acids, which then get collected into the gallbladder. And then, at the need, when it's needed, they get pumped, secreted into the small intestine. The pancreas is another gland which has both an endocrine and an exocrine function. The most important exocrine function is the one of producing digestive enzymes, but also it has very important related to digestion endocrine function, which is to secrete insulin and glucagon to maintain the homeostasis, the homeostatic levels of glucose in the plasma. The homeostatic level of 100 milligrams in 100 milliliters. So in the pancreas, we have basically two glands which are mixed into one. Okay, so we do have the acinar cells of the pancreas, which secrete digestive enzymes, and these are amylases, lipases, and proteases. And these epithelial cells, these acinar cells, secrete the digestive enzymes and bicarbonate into the small intestine via the pancreatic ducts. But then inside of the pancreas, within the pancreatic parenchyma, we do have these pancreatic islets, which contain two kinds of cells, alpha cells and beta cells. And these islets, which are enclosed within the parenchyma of exocrine acinar cells, these alpha and beta cells are responsible for the synthesis and secretion inside of the plasma circulation of two hormones, insulin and glucagon. As you know, insulin has the function to decrease the blood levels of glucose by promoting the absorption of glucose by muscles and liver, and into liver and into muscles promote this synthesis of glycogen, while glucagon has the function to increase the plasma levels of glucose by promoting the conversion of glycogen into glucose from the hepatic cells and the release of glucose into the bloodstream.